A very good evening, students. The title of this lecture is Sex Chromatin and Lyon's Hypothesis. Now, this is an important, short note question which is very frequently asked in the exam. Okay, so I move to the next slide, and first we will learn about the sex chromatin, and then I will tell you about the Lyon's hypothesis. So moving to the next slide. Now we come on to the sex chromatin first, okay? Now in humans, sex can be determined by the just looking at the nucleus of an interphase nucleus, okay? Just looking. Interphase means the resting nucleus. In the last lecture, I showed you the cell cycle, okay? And cell cycle is was consisting of the two component interphase component where the cell was resting, and second part of the cell cycle was mitosis, okay, where the cell division was. So it is a large duration where the cell is resting and then it goes to the cell division. So when a cell is resting means when it is in the interphase stage, okay, and if you observe the cell at this interphase stage, then you can see a dark staining chromatin mass. And what is the chromatin mass? It is the chromatin was the nuclear material inside the nucleus which we have divided into two. The dark was called as heterochromatin and the light chromatin mass was called as the euchromatin. So in this case, when you are looking at the nucleus of an interphase cell, somatic cell of course, then you can see a dark staining chromatin mass which is attached on one side of the nuclear membrane. See this is a cell, a somatic cell, and this nucleus is a mixture of hetero and euchromatin, okay, heterochromatin and euchromatin. And then you can see here, see this where my pointer is there, there is a dot like structure which is attached to the nuclear membrane on one side, okay, and this dot like structure which is heterochromatin or dark staining chromatin material which is there, this is called as the sex chromatin. This is called as sex chromatin. And this sex chromatin since was discovered in 1949 by uh, one professor of anatomy, that means who was teaching the neuroanatomy in Canada, in London of Canada. This is different London than the London of UK, okay, it is in Canada. So he was teaching the neuroanatomy, and while doing some research onto the brain, and probably it was cat brain, he was seeing the slide of the brain of the cat, okay, under microscope, and he just observed this. That means whenever he was observing this slide of a female animal, okay, then he was always observing this kind of the chromatin mass attached to the nucleus. And since it was just discovered by Barr, okay, Professor Barr, okay, so it is called as the Barr body. Now, but this he also observed that this was absent in the males animal, okay, when he was observing the slide of the male animals of the brain, that means he was observing neurons, nucleus of the neuron which is mostly euchromatic, and that's why he could observe these, okay. Why he could observe this bar body? Because the nucleus of the neurons, now cell, they are light stained, euchromatic, and most of the chromatin material, it is extended, okay, not tightly coiled. It is pale nucleus, okay, and on the one side of this pale nucleus, this bar body or chromatin material was very much visible. Now suppose if he would have been observing some other nucleus from some other part of the body or tissues of the body which are not as euchromatic as a neuron, then it would have not been possible for him excuse me, to observe the Bohr body, because most of the nucleus in other tissue, they are a mixture of heterochromatin and euchromatin, where heterochromatin predominates. So he was lucky because he was looking the nucleus of a neuron. Okay. Then he also observed, as I said, Bohr body was absent in the male animal. So whenever he was observing the slide of the brains of the male cat, probably, I'm not sure which animal he was observing. Okay. So this was his observation. Okay, it was in 1949. Okay, and since he was the professor of anatomy, all the time he was engaged in teaching the neuroanatomy and its research. But soon after this discovery of the Bohr body, all of a sudden he became interested in genetics, and then his line of research changed, and he was just only doing the research on genetics, cytogenetics. Okay, but teaching is still teaching the neuroanatomy. Okay, because he was professor of neuroanatomy, and he became very successful, got so many awards. Okay, right. So this is how the Bohr body was discovered by, or the sex chromatin was discovered in 1949 by the Bohr. He never bothered to research further on the Bohr body itself, as I will tell you after a few slides. Okay, so then other observations by some other people, not by Bohr, okay, was that this kind of a chromatin mass was also seen in the nucleus of female polymorphs. Okay, female later on. Okay, white blood cells of the females. When a person is observing a slide of the blood, then if the slide is from a female, then this kind of another structure was seen on coming out of the nucleus, and this was thus its shape was like a drumstick. So this was also, so drumstick was seen in the nucleus of female. So Bohr body or sex chromatin and the drumsticks, they are only present in females. Okay, they are only present in female. Right. So I move to the next slide. Now we learn more about the Bohr body or the sex chromatin. Now the identification of the sex chromatin is thus become an easy and quick method to determine the sex of an individual. Later on, after animal, uh, people have worked out, and Bohr himself has worked out that it is also present in human being. Okay, sex chromatin or Bohr body. Okay, or the drumstick was present in the females, human females. Okay, so it become a very quick method. That means you most of the I will tell you the procedure later. Just let me read this. Most of the somatic cells of a female, somatic cells, I have told you many times, those all the cells of the body of various tissue except the sperm or ovum, they are the somatic cells. Okay, they are called as gametes, which have the different genetic constitution. Okay, uh, the somatic cell if it is obtained from the oral mucosa, from the cheek, okay, or from vaginal mucosa, okay, or epithelial lining. Mucosa, you will learn later when you will go to the histology what is mucosa. Okay, the vaginal epithelium or urethral epithelium, or from skin, okay, skin cells, yeah, epidermal cells of the skin, or the blood cell. There will be drumstick in the blood cell. Okay, so these are the cells, somatic cells, where you can very quickly identify the presence or absence of the Bohr body. Okay, and these are used to identify the chromatin, uh, sex chromatin. Now the procedure is that you just with the spatula, wooden spatula, just take the scraping of the oral cavity, that is from the cheek of the oral cavity, and then this is sprayed onto a glass slide, and it is fixed in alcohol, and then it is stand with any of the basic dye. Even hematoxylin is good enough to stand these cells, okay, which are on the slide, right? And then you observe this under the high magnification, okay, because in low magnification nucleus will not be large enough to see the chromatin, okay, sex chromatin or bar body. Then you just move the slide from here to there and observe in many cells, okay, this bar body. So cheek cells, the means say epithelial cells from the oral cavity, okay, will, if it is from female, they should show the bar body or the sex chromatin attach onto the inner aspect of the nuclear membrane, a heterochromatin mass or heterochromatin mass, okay. Then if you observe in few cells this presence of this bar body, then you are sure that the person from whom this scraping was taken is a female. Okay, it is. So you have determined the sex just very quickly within 5 minutes. Okay, within 5 minutes. But the determination of the sex by bar body is not very satisfactory. It may so why it is not very satisfactory. It may so happen that even if it sees female, then it may so happen that you may not observe the bar body, because bar body is just a dot which is attached onto the nuclear membrane inner side, and it is by chance it is seeing, okay, if you are seeing in that direction where it is present, okay. If, see, imagine that nucleus is a spherical ball and a dot on the inner aspect can be seen only luckily, otherwise it may be hidden, okay, from that. So only few cell if you are showing, and it may so happen that even not a single cell may show the bar body even if she is from female, okay. That is the practical problem. So it is not a very satisfactory method to determine. But on other hand, karyotyping, the chromosomal analysis, as we have seen, okay, it is an accurate, because there you are seeing an exactly two X chromosomes, okay, which will identify the sex. And if X and Y are seen, then it is from the male, right? And in case of thus male, you are not sure if you are not seeing the bar body, so many cells you are observing, and if it is not seen, then you can say that bar body is not seen and probably it is male, but with certainty it is difficult. But yes, if you have seen the bar body in the under the slide, you can say that person is a female, okay. Now this is a quick method just to know what is the sex of a person, and this is used in Olympic games, okay, just as a routine test to see the sex of a player, okay. Some player may hide their, I mean to say, sex. They may be male but pose as female, okay. So to win the game, and there it is a routine too. And the first method by which the sex is determined quickly is this bar body, okay, or sex chromatin analysis by taking the scrape from the oral cavity. Now, then a big question comes, why bar body is present in females only but absent in males? Okay. As I said that the bar, professor of anatomy who discovered the presence of bar body, never thought in this line. Okay, must have thought but could not succeed. Okay, could not succeed. The bar body is present. Why it is present only in females and not males? But at the same time, after 10 years, and another group of scientists, they found it, okay. In 1959, it was discovered that the sex chromatin is derived only from one of the two X chromosome. Females contain two chromosome, X chromosome, okay. Out of these two, one was responsible for formation of the bar body, okay. Out of two X chromosome, one becomes condensed and thus become inactive, okay. That was told in 1959, that out of two, one of the chromosome is inactive, and that's why it is condensed, and I mean say it is condensed, that's why it is inactive, and thus it is attached to the inner surface of the nuclear membrane, okay, in the form of the bar body. The other X remains active, thus it is euchromatic. Okay, it is expanded, and because the genes present on these X chromosomes are constantly active. The other one is condensed, hence it is inactive. So gene presence are not active on that uh, X chromosome which has formed the bar body. Okay, this could have been very well observed by the Bar himself, which was discovered after 10 years. Okay, but the luck, I will say that he must have not made these efforts on this side. Now, as the males have only one X chromosome, because the other is Y chromosome, very short Y, only one X chromosome, it remains active throughout the life. I mean, say it always remain active, and thus bar body is not formed. Okay, this is the reason why females they have the sex chromatin and are bar body, and males they don't have any bar body. The reason is that, okay, the reason is that males have only one X chromosome, and this is necessary for the life. That's why this X chromosome is always in euchromatic form, and it is active. Genes are active on this. Okay, so this was discovered in 1959. Let us see further. Okay, then soon after, 3 years after this discovery, when one said that one of the X chromosome become condensed and inactive and forms the bar body, soon after that, a big hypothesis comes, and it was given by Lady Mary Lyons. Okay, Mary Lyons. Now, this lady said that in females out of two X chromosome, any one will become inactive. This was not told by previous investigator who said that out of two X chromosome, one is inactive, okay, and forms the bar body. She says further, he further analyze what they have said. In female out of two X chromosome, any one becomes inactive. Okay, anyone means there are two. Out of this, anyone randomly one will become inactive. It is not that this particular chromosome, one which comes from mother will become inactive, or one which comes from father will become inactive. Randomly, anyone out of the two will become inactive in early embryonic life. Okay, she was pinpointed where at what stage after fertilization this X inactivation or formation of bar body will take place. This was on the 15th or 16th day after fertilization. 15th or 16th day after this big work. Okay, she must have worked a lot to just find out on which day the bar body is formed. Okay, in an embryo, and then it forms the bar body after inactivation on 15th. After 15th and 16th day, if you will see any embryonic cell before 15th day, no bar body will be seen even if she is female. But when you will observe any embryonic cell after 16th day of embryonic life, definitely the bar body will be seen. Okay. Thus, as inactivation, this X inactivation, okay, when where one X chromosome become inactive and form the bar body, condense and form the bar body, is called as Lyonization or Lionization. This is called as Lionization. Okay, the process of inactivation is called as Lionization. Okay, I hope you have understood it. Now the X chromosome to form bar body is randomly selected, as I just said. Either that X may be from mother or that X may be father, from father. It means that some of the X chromosome in some cells becomes inactive, which are from mother, and in other cells, okay, this X chromosome from father may also become inactive. Okay. But this is very important, we see discovered, is that once a selection is made that which X chromosome will become inactive, whether it is from mother or from father in a one particular cell, then afterward all the daughter cells, okay, will be will become inactive for that X chromosome only. That means if in a cell, if the X chromosome for the mother has become inactivate, then in all subsequent cell division the X chromosome from mothers will get inactivated. Okay, in all subsequent cell division. It is not that in the next cell division, okay, the X chromosome father will become inactivated. Okay, it's not like that. Okay. Okay, so what does it means? So it means a female body, somatic cells in female body, they contain two cell line. Two cell line means some of the cells are in the female body, out of the billions of cell, some of the billion cell, 1 or 2 billion cells, say for example, may be having the inactivated X chromosome derived from mother, and other cells, remaining cells may having the X chromosome derived from father, which is inactivated. Okay, I hope you got it. The number of bar bodies is always one less than the number of X chromosome. I will up the point which I just left, that one selected it will remain the same. Okay, this I will explain you by a diagram. Just this is also important. This point you just understand. The number of bar bodies is always one less than the number of X chromosome. That means if a female since is containing two X chromosome, one will become inactive, okay, and another will be active. So if a female who is suffering from Turner syndrome, where she has only 45 chromosome, out of which only one X chromosome is there, that's why one chromosome is short. So since she is not having two X chromosome, only one, there will be no bar body. That means no inactivation of X chromosome will take place. The only one X chromosome which is present in somatic cell, that will be always active, euchromatic. Okay, and if a person who is having two X chromosomes, say for example XXY, which is Klinefelter syndrome, okay, the sex is male, the person is male, okay, and but he is having two X chromosome by non-disjunction, okay, which I will tell you after 2 or 3 lectures. This the male is having two X chromosome, so there will be one bar body, okay, and one X and Y will be active, okay, one X and one Y will be active, and another X which was axis, in axis it will become inactive. Now if a female is having 3X chromosome instead of normal 2X chromosome, and this kind of the females, they are sometimes called as super females, which are having one extra X chromosome. Here the two bar bodies will seen, and only one X chromosome will remain active. So two bar bodies will be seen, right? Two X chromosome will become inactive. And in an XY normal male, which is having only one X chromosome and one Y chromosome, there will be no bar body. That's why we don't get any bar body in males, okay, because there is only one X chromosome which is always active. Now I quickly move to the next slide. This is that diagram which I was referring, that how the inactivation takes place. So see that this is an embryonic cell where two chromosomes are seen. One is in red and another is in the green color, right? And one where it is written as M is means this is the X chromosome coming from mother, which has come from mother, and another P written, it has come from the father, okay, maternal or paternal chromosome, X2X chromosome. Now it is an early embryonic stage, okay, and repeated cell divisions are taking place, okay, and then this cell division will keep on taking place, and there will be no change here. But when 15th to 16th day of embryonic life will approach, then on the 16th day or 15th day, excuse me, there will be an random X inactivation will take place, okay. That means on that day one of the X chromosome will become inactive, okay, or condense and will form the bar body or sex chromatin. Say here in this cell, okay, this red color chromosome, X chromosome, which has come from maternal uh, or from mother, it will become inactive and will form the bar body. And then later on in all subsequent cell division, thousand, hundreds or millions of cell division which will follow for this cell, always the maternal chromosome will remain inactive. The paternal chromosome coming from father will always remain active, okay, in all the subsequent cells. See here what happened here, that there is the maternal chromosome is active and paternal chromosome become inactive and it has formed the bar body. So in all subsequent division, cell division, there will be millions of cell division will take place, because from embryo and adult body formation and somatic cell division keeps on going, okay, keeps on going. So in this way, one of the chromosome, X chromosome, either coming from maternal or paternal, randomly it gets, I mean to say, inactivated, okay. Now in the other half, again the similar thing is shown here. Here you can see that the maternal is active in both, and then subsequent divisions only maternal X chromosome will remain active and the paternal will remain inactive. Okay, this is just to show you the subsequent cell division will have the same chromosome inactivated all the time. Right, moving to the next slide, and this is the last slide, okay. And now the question come, why an X chromosome become inactive in female, only in females, okay. Why an X chromosome becomes inactive in normal females, I will say, and where in axis they are present, there also. Now this phenomena of inactivation was now explained by this, I mean to say, Lyons, Mary Lyons. She said that it is due to the dosage compensation. Okay, dosage compensation. We will understand what is dosage compensation. Now, explanation is this, that all female cells contain two X chromosomes, okay, while male contain only one X and one Y. Yes. However, the level of protein product of X chromosome is same in males and females. What is protein product of X chromosome? That means genes sitting on the X chromosome, okay, which determines the female sex, they keep on producing, because genes function is to produce the protein enzymes, okay, uh, by sending the messenger RNA, okay, to the cytoplasm, where endoplasmic reticulum, where the polypeptide chain will get condensed, and then it goes to Golgi body to secrete the product. Okay, so genes are involved in the production. Okay, so the level of this product which are produced by the genes on X chromosome is same in males and female. Okay, so both the sexes, because both are now having the one active X chromosomes, that's why the product are same. Okay, now, this is because in female there were two X chromosome, one is inactive as early as 15th day of embryonic life, okay? One is inactive. That's why both are having the same quantity of the protein produced by the X chromosome, okay? By the X chromosome. So if both the X chromosome were active in female, now think a situation, hypothetical situation, you think, if suppose in female both the X chromosomes were active, then the product of protein or any product of the, now I mean to say gene, will be in double dose in the females, because there are two X chromosomes and males are containing only one X chromosome, so it will be in double dose, okay. Hmm. And this double dose of this protein, which may be in the form of hormones, enzymes, or any chemicals, it will be lethal. That means it is not. It means an extra X chromosome, numerical chromosomal abnormality will, a condition similar to the numerical chromosomal abnormality will take place. So it is not good for the health. Probably the female will die, okay, if two are active simultaneously. Thus, for this reason, one X chromosome becomes inactive in females. And how it become inactive in females for throughout the life? This is by formation of the sex chromatin or bar body where it is tightly condensed, okay? So that no gene can act which are present on this X chromosome. Thus, this is called as dosage compensation. Sometimes a short note can be there on dosage compensation, right? So this is the answer for the what is dosage compensation. Now you have learned, yes, there should be two chromosomes. One should be active so that male and female, they should produce the same type of the, I mean, say same quantity of the proteins, right? But what about an extra chromosome in male which is there? The counterpart of those genes in which are present on Y is not there on the X chromosome or anywhere in the whole karyotype of the uh, females. The reason is that nobody bother, I'm saying there is no need to bother for the compensation of the genes which are present only on the Y chromosome in a male, because Y chromosome is very small. It belongs to the last group of the this classification, okay, structural classification, the inverse classification. And on this small Y chromosome there are very few genes, and these few genes are just mostly determining the sex, male sex, okay, and it has nothing to do for the female. And few genes which are there, they are mostly for the secondary sexual characteristic which is male type, okay, male sex secondary sexual characteristics. And those some of the genes which are for the mustache and beard, okay, or distribution of pubic or axillary hair in the males, which is not very important function as compared to the production of the proteins which may be lethal, which are present on the X chromosome. That's why Y chromosome dosage compensation is not needed in the human beings, okay, it is not needed in the human. Now one more thing I will like to tell you a little, this lecture is going a little longer, but I will tell you one more thing. Thus a female is what? She is a mosaic. Mosaic means a person who has two different genetic constitution. That means the two different kind of cells a person possessing. I will tell you more about the mosaicism when I will teach you a separate lecture on this, okay? So a female is a mosaic. There are two cell lines, two different genotypic cell lines are there. Why there are two different genotypic cell line in a female? Because some genes are from mother and some genes are from father, because in some cells the maternal X chromosome is active and in other cell the paternal X chromosome is active. Thus she is considered as the mosaic in one sense, okay, not in very true or strict sense, right? This complete this lecture. Thank you very much.